All right, so thanks everyone for taking the time to come in. I really appreciate it. And what we wanted to show you today is kind of what we are planning to develop. These workshops are supposed to work as a practical, hands-on
This is a
exercise
demonstration of
focused on a specific
Webcast.
type of omics data. So the first one that we started with is transcriptomics, but one of the links that we gave you actually has a website that kind of describes briefly all the different types of omics data and then integration, machine learning, and that's kind of where we're going with this. This is our first workshop, and so we're trying to also learn from you. So if you want to let us know how it went, what you liked, what you didn't like, and how you would like us to improve it at the end, we would really appreciate it.
This is a demonstration
Claudia
of
here
Webcast.
is going to start with her part of the presentation.
And I might add, we'll send you, in order to save time, because this is a lot of information compressed into a short time, we'll send you a kind of post-evaluation, send you the pre-evaluation sent out, where you can tell us and all right, so we are going to be talking about the BioInfo platform, and the platform is capable of many different types of analysis.
This is a demonstration of Webcast.
Genomics, it can do transcriptomics, proteomics. There's specific metabolomics. There's CIRSE, which is a specific sort of virology program. But what we're going to be looking at today is RNA analysis. And the name of it is RNA-seq. So before we go into what RNA-seq is, just a brief review of what RNA is. So we're looking at all RNA
of
in the cell.
Webcast.
So messenger RNA creates protein. Protein functions based on its shape. RNA, since it's single-stranded, can also fold into a shape. That's called non-coding RNA, and it works directly the same way that protein does. So many different types of non-coding RNA. tRNA and rRNA are really familiar. These other ones are less familiar, but they're really important. They involve things like defense and expression regulation and all kinds of different functions. So another brief review. In the genome, we have exons and introns. Introns are spliced out, so that's what you're going to get in the mature RNA transcript. The thing is that splicing can be done in different ways. So, for example, in this gene, you could have these introns spliced out, and all 4 exons end up in the final transcript. In this one, you have this intron spliced out, but this one, it's going to go from the edge of this intron to the edge of this intron, splicing out that full exon. So you end up with a final transcript that's got 3 exons that's shorter than this transcript. These two things are called isoforms.
This is a demonstration of Webcast.
Some isoforms that could be not just shorter or longer, but you can have completely different isoforms, as in this case, where one is E1, E2, and E5. This is E2, E3, and E4. So you have different types of isoforms. The data that we're going to get is from this end. So we're looking at the transcripts, and then we're going to take the transcripts and then go back to a genome and place them on the genome and find out where the introns are and other information.
This is a demonstration of Webcast.
Oh, and non-coding RNA also has introns. Some of them do. They're long enough, like X chromosome inactivation. So, why do we want to do a whole transcriptome analysis? It allows you to look at unknown correlations, and this is really important because there's a lot of questions that you might not have a hypothesis. You might be approaching without one, such as you have a disease and you've got a drug and it's 70% effective. That means 30% of the patients are not responding. Why are they not responding? Maybe
this is a demonstration
of
Webcast.
You can, rather than go through the process of building a hypothesis, you can go through and just do a whole transcriptome analysis. You compare the responders and non-responders, and you can immediately come up with a lot of relevant information. In the absence of a hypothesis. There's also disease correlations with post-transcriptional events, and species without a well-annotated reference genome. You can also use this for that. We won't today, but you can. And power. It's more powerful than microRNA.
This is a demonstration of Webcast.
Large-scale RNA analysis. So, in microRNA, as I think probably most of you know, you have a gene chip and you have several probes put on that gene chip, and you probe them, and the fluorescence the RNA fluorescence labeled transcripts shows up, and the degree of fluorescence is correlated with the gene expression. So brighter fluorescence means more gene expression. With RNA-seq, you're really looking at it directly. So you're fragmenting the entire genome, looking at the number of fragments that line up with each gene. So your expression level is going to be the actual fragments that you see that are expressed for that gene. And this has a number of advantages. One is that you have the actual sequence of this, as opposed to just a fluorescent signal where you know the expression of something is higher. You can see the actual strandedness and other information about those transcripts themselves. It also allows you to easily do
This is a demonstration of Webcast.
So, and it also has a lower error rate, and you don't have problems like cross-hybridization. But really, the best thing about this is that with a microRNA, you have to decide in advance what you're going to put on the gene chip. So a lot of times it's maybe expressed genes. But you're going to miss things like non-coding RNA that might be important, but you just don't know anything about it yet. So with RNA-seq, you're doing the entire transcriptome in a sense in a
This is a demonstration of Webcast.
And then you can move it from there. So the first step to doing an RNA-seq is to the sequencing itself. So we need to use next generation sequencing. So this is high throughput sequencing, and this is for DNA, but we're doing RNA. So the first thing you want to do is fragment the RNA, then you want to first transcribe the RNA into cDNA, and then you do high throughput sequencing of the cDNA. These fragments are going to be, I'm going to put it in the back on the end, and then these allow them to be attached to this flow cell, hybridized to that, and then they're sequenced off of these flow cells, and you get a huge amount of sequencing there. So thousands and thousands of reads. Now a read, a couple of other things. There's a variety of machines to do this. So some machines are much more powerful than others. You have machines that can give you longer reads, that can give you more reads per run. Which you use is going to be based on cost, but also experimental design. So if you go to Illumina, they'll have charts asking you about what experiment you want to do, what's your priority, so that you can choose the best machine for your particular experiment. So when you get an RNA-seq read, this is actually a sequence read of a fragment, not of the entire transcript. That's an important point to remember. So the fragments are sequenced. You can sequence it either one way, that's called a single end read, or both ways, that's a paired end read. And this, among other issues, the length of your read is going to affect your analysis. So imagine if you're trying to build a puzzle that's a certain size, and you have that puzzle cut into 100 pieces versus 1,000 pieces. You know what's going to be easier for you to solve. So the solving of the puzzle is basically done by your software. So that's on the software interface.
This is a demonstration of Webcast.
Is done by your sequencing interface. So if you have a more powerful sequencing machine, you can have longer reads, but you also have issues like higher error rate and so forth. And the software that we're using is quite powerful. So it kind of precludes the need for really long reads. So the other issue with preparation of these reads is that you need to clean counters from your library. Now, once you have these done, you're going to align them to what's called a GTF.
Demonstration of
GTF just means that it's annotated, so the genes have the name of the gene, like that sort of thing. So as these fragments get aligned, you can see that certain fragments are going to have, the fragment is going to go from here to here, but on the genome there's space in between them. This is the intron. So you can see with all these different fragments lined up, there's a clear intron here in all of them. Some of them, though, there's a longer intron and some have a shorter intron. From all of this data together, these can be assembled into transcripts. So based on these orange ones form the most abundant transcript. So you've got 3 exons with these small introns. This is a little less abundant. You've got a longer intron here. and the least abundant has skipped these exons altogether. You have another exon down here, long intron, and then these 2 exons. So when we talk about the transcripts being assembled, that's what we're talking about. Okay, so that is the step 2, mapping onto the transcriptome. So you're taking your reads, you're mapping them onto a genome. For this, for the sake of today, mapping onto the transcriptome is just like mapping onto a genome, but with the expression levels. To do that, you have a choice of different programs that can do that for you. The algorithm we're going to be using is called BowTie2. So you can do this with free software. You can load your data in, and then when you take the output of that, you feed it into another program, and then you can take that output and feed it into another
Webcast.
At each of those steps, there's a chance of error, and it has to be formatted correctly. It's somewhat difficult to use. What BioInfo does is it makes this very easy. It's just a button. You just click on the button, and it does it, and it's ready to put the data into the next step, which in this case is step 3, generating expression tables. So these are going to be tables of the genes that your fragments map to, and then isoforms, which are the different versions based on alternative splicing.
This is a demonstration
So
of
the program we're
Webcast.
going to use for this is RSM expression table, and how we are going to tell the system how to do that is to use the BioInfo platform. So there are many different algorithms here. You can do all kinds of different experiments, customization. You start here, and then you pick the different algorithms that you want to run. So in this case, it's going to pick this algorithm, top hat goes to this one, cufflinks to bow tie, going to here and going to end.
This is a demonstration of
What we are
Webcast.
going to do is a very simple one. So we're just going to start with our data. We're going to do BowTie2 to map on the transcriptome. We're going to get an expression table with this RSM expression table, and then go to end. And all we have to do is just click, 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 and end. And you have what's called a pipeline. That's a pipeline of algorithms, basically a chain of algorithms together. Okay. So now, at the end of the pipeline, now what are we going to do?
This is a
Have a table of
demonstration of
transcripts.
Webcast.
First thing is normalization. Jacqueline's going to talk a little bit more about that. There's many different techniques. You want to make sure that you're not reporting results that are due to technical issues and those issues not related to your experimental question. Post-processing. So principal component analysis, I'm going to have another slide on this. It provides kind of a visual overview of your data. You can do statistical analysis, and then machine learning techniques.
This is a demonstration of Webcast.
And then the final step is your biological interpretation of your results, and you're using all the tools that you already know how to use, databases, publications, and looking at these results and seeing what their biological meaning is. So PCA, it's a dimension reduction technique. So what this means is when you get your output, you're going to have a huge table. There's thousands of genes. There's nothing that you can do as far as looking at that table to find the meaning of that data.
This is a demonstration of
So what we want to
Webcast.
do is reduce this down to sort of a snapshot that's somewhat meaningful. May not capture everything in the data, but it's a meaningful snapshot of the data. So it's analogous to a shadow. So if you take a three-dimensional object, you shine light behind it, you get a two-dimensional representation in terms of the shadow. And that is my fault. So sorry. Oh,
This is a demonstration of Webcast.
So just as you can reduce a three-dimensional object to a two-dimensional representation, which is meaningful, may not capture all the detail, it is meaningful, we can take a 3,000 dimension object, if you want to consider the data set an object, and reduce it down to 2 dimensions, and that's what PCA is. So this is the picture of what you would get. Each of these points is going to be a sample, and it is all of the RNA sequence information for that sample, the entire transcriptome, reduced to one point.
Is a demonstration
And you
of
can see in
Webcast.
this picture that these points cluster together and these points cluster together. So you can see there is a difference between these types of samples and these types of samples. Then you can go into your data and find out what the nature of that difference is. So now machine learning was the other post-processing thing I mentioned. And this is the use of what's called an unsupervised analysis. So say you have a problem where you want to tell the computer to tell the difference between two things. You can tell it what to do and say, okay, look for things, and this example is going to be ducks. So if you want to say this is a picture of a living duck, this is a picture of something that's not a living duck. Okay, look for things with beaks. Well, there'll be some exceptions to that. Look for things with feathers. Well, there'll be some exceptions to that rule as well. So an alternative approach is to simply feed into the computer, so 10,000 real ducks, 10,000 things that are not ducks. And the algorithm itself will figure out the best way to separate those groups. Now that it has separated those two groups based on its own criteria that you're not given to the computer, it's what's called unsupervised, you can now take unknown samples and take a look at whether they're likely to be a duck or not. So this sample falls firmly within the real duck range. We're 99.8% sure it's a real duck. This one falls pretty well into the not Wirecast range. So 97, 98% sure that it's not. And this one we've got an 86% sure that it's not a duck as well, because it's falling within this clustering. So you end up with an output, and in this case it's correct. Why do we want to do this? These graphics were actually created by a web security company. So when you're trying to identify hackers and criminals and so forth, you can't just tell it, okay, this is what the criminal will do. Detect this behavior.
This is a demonstration of Wirecast.
So instead, if they can do a training set based on what normal people tend to do on the internet, then what some known criminals do on the internet, they can create flags. They can say, maybe this is a 70% chance that this might be criminal, and the guy calls you up and says, hey, there's some unusual activity. Is this real? So this is, when it comes to genomic analysis and transcriptome analysis, we're also dealing with a huge unknown set. So when you're dealing with such a large set of unknown data, machine learning can be extremely useful. Okay, so what we're doing today, Jacqueline is going to guide you through an actual RNA-seq project looking at breast cancer. So we're going to look at 3 types of breast cancer. One is estrogen receptor positive, so it expresses the estrogen receptor. So it's you can have specific treatments for these subtypes. So this is going to be a hormone-oriented treatment. HER2 is expressing a growth-promoting protein. Also has specific treatments for that type of cancer. Triple negative doesn't express progesterone receptor, ER receptor, or HER2 immune receptor. So they it doesn't have a specific treatment, but it's still useful to know because you can make informed decisions based on that. The other thing that Jacqueline is going to be talking about is PDX models, patient-derived xenograft mouse models. This is basically taking a tumor from a human and implanting it into a mouse. So there's a couple different issues with this.
This is a demonstration of Wirecast.
The mice do need to be immunocompromised, or they'll reject the tumor. The way that they're immunocompromised is relevant to the experiment. So an aponic nude mouse model is going to be unable to produce T cells, but can produce all the others. Non-SCID will have no T cells, no B cells, but they'll have natural killers, macrophages, and granulocytes. So you need to consider the type of mouse whenever you're making drawing conclusions based on
this is a demonstration of Wirecast.
The other terminology that Jacqueline's going to be using is tumor and stroma. So stroma is the microenvironment surrounding the tumor. It's initially going to be human. When they cut the tumor out, they're going to then implant it into the mouse. It's got human tissue around it. That tissue will die, and the mouse tissue will take over. At the point of the experiment should be that all of the stroma is mouse tissue, and all of the tumor tissue is human tissue. So
this is a demonstration of Wirecast.
So that way when we're going through this, everybody's got everything they need. So the email that David sent, you're going to want to have that in front of you. There is a link there that will take you. go here. Yeah, so this link, the EDUT BioInfo link that's in the email from David, you want to click this, and when you get there, we want to go ahead and open up these links here. So you've got links for principal component visualization, factor analysis, ensemble, and David. So I'll just let you guys get to your emails, and we can help you with that. Will this be helpful? And then in that same email from David,
this is a demonstration of Webcast.
Got them.
This is a demonstration of Webcast.
One thing we also found is that bypass is a little bit finicky, so we recommend that we use
Well,
my only
This is a demonstration of Webcast.
I'm curious. So we had 5 people submit for the pre-evaluation. But I think it's kind of want to get a general idea. Has anybody ever done RNA analysis or worked in that kind of field?
This is a demonstration of Webcast.
So we're going to go through this. It's kind of in the middle level as far as difficulty. Somebody might be bored for a little bit. Somebody might find it really difficult. So just as we're going through, if you find anything, or you start getting lost, let me know when I say them. David sent an email last week, I believe, that had the link for the paper. So the paper that we're analyzing today is whole transcriptome profiling of cancer tumors in mouse fetus models. Absolutely not required to have been read before today.
This is a demonstration of Webcast.
I'll go over the introduction for the paper, but probably we've already talked about it a bit. The breast cancer subtypes, why would we care about subtypes? If you yourself develop breast cancer, it's very important. If you have been diagnosed with triple-negative breast cancer, the common drugs, which are basically suppress the hormones, don't work for you because those cancers don't rely on hormone receptors, they don't have them. Estrogen receptor
This is a demonstration of
this
Webcast.
is the most common, easiest to treat. If you did get breast cancer, you probably want to do that version, but you wouldn't want it at all. And then the last is HER2. So it grossly overexpresses this human epidermal growth factor, and it allows it to just proliferate wherever it's at, so grow really fast and replicate, which is unfortunate and has to be slowed down with drugs. Those subtypes are in the experiment itself. They took 21 different samples from actual patients.
This is a demonstration of Webcast.
And then they let the tumors grow within the mice over a number of weeks, somewhere between 8 to 20. And this is important because what the paper was focused on was to look at what the interaction was between the stroma, so that microenvironment that forms around. Okay. Oh, we have a few more. Let's see if we can get some chairs for them.
Oh, yeah, right.
Welcome, guys. Thank you
This
for
is a
coming.
demonstration of Webcast.
Yeah, no problem at all. Thanks for coming. Julia, how did you type in the product? Yeah. So we have.
You want to grab this one?
Hello.
Yeah.
guys? So we'll kind of help
This
you
is a
get
demonstration of Wirecast.
set up for what I'm doing. I'll just kind of go back into the intro a little bit. Yeah? Okay. So the paper that we're going to be doing analysis for today is whole transcriptome profiling of cancer tumors and mouse PDX models. I sent the paper last week to David, but we forwarded it over to you guys, but it was not required to you guys to understand today. It would have been helpful.
This is a demonstration of
Just a
Wirecast.
brief introduction. Again, sorry if you guys are already in here. There's a number of breast cancer subtypes that were used in this study. Why would subtypes be significant? If you yourself have breast cancer, it's very important. These subtypes differ in their expression, so triple negative cancer doesn't have hormone receptors overexpressed on the outside of that cell. So it doesn't require estrogen to grow, which means most of the therapies, like endocrine therapies,
This is a demonstration
so
of Wirecast.
And no one diagnosed that subtype, they would probably give you the wrong type. The most common is ER+. This is an estrogen receptor positive breast cancer. This one has the most positive outlook. If you're going to get cancer, it's going to be want and then HER2. So it overexpresses a hormone, human epidermal growth factor, and it makes it more aggressive. So it grows faster, proliferates faster, and there are drugs.
This is a demonstration of Wirecast.
The paper has over 80 samples. They look at 7 different cancers, all different types of subtypes. We have you guys print out half, so we couldn't do it. So we're going to just look at the breast cancers, 21 total samples, and 50 cancers. So if I look at just the Excel table for the sample summary, we have run numbers. These are assigned whenever you actually take your data and submit it into a biological
This is a demonstration
database.
of
So
Wirecast.
These are the most common. Some of you are probably familiar with it. NCBI, or the National Center for Biotechnology Information, and EDI, or the European Molecular Laboratory. If I take a look at what actually looks like on the web page, this is the data set for the paper that we're looking at today. Normally the names will be similar to what the paper is. There's an SRA experiment, so this is where you go to download your samples.
This is a demonstration of Wirecast.
You find a paper that you're looking at that you find super interesting, and you want to know if you can access the data. Commonly in the molecular biology field, you would look for an accession number in that paper, and you would normally find it on NCBI and EDI. So these say the same thing, just in a different version. So you can look through Express or Array Express to see different variables for each of these samples. For us, if you want to do my pipeline, you need to download the FAST.
This is a demonstration of
What is a
Wirecast.
FAST file? So a FAST file is large. It's going to have this set of 4 lines for every single read that maps onto the genome. So I'm giving you an example of just one of those reads. A single read in the FAST format looks like this. So the first is just a name. It's arbitrary if it comes from the type of sequencer that was used, the experiment. The type of researcher may have his own naming. The second, very important line,
This is a demonstration
so
of
this
Wirecast.
is the base call that was predicted by the sequencer itself. The third is a placeholder, you can ignore it. And the fourth are the base qualities. Yeah. The base and the quality line up. So each of these quality scores is corresponding to the above nucleotide. And if you were a sequencer and you're trying to think of it in that way, what does it actually mean? So here, if I needed to call a base,
This
and
is a
by that,
demonstration of Wirecast.
and say, what is it of the 4 nucleotides, it would be very easy here, because we would have high quality scores. If I needed to guess here, you might not know what that is, right? Because you have 3 very high peaks that are overlapping each other in the same spot. So there you have a low quality score. And that's important. FASTQ is the standard type of output 4 high throughput data sets. And there is one other format which is just FASTA.
This
and that
is
has
a
just
demonstration of
no
Wirecast.
quality storage associated. So Bonnie mentioned a bit about the pipeline that you're going to build, so I'm not going to go into that. What I will say is that we are mapping on a mouse and a human genome combined. Why? That has to do with the experiment. So in the experiment, they put the tumor in the mouse. They didn't try to separate the RNA. They just sequenced everything. So you have both the mouse stroma, or the microenvironment.
This is a demonstration of Wirecast.
Those will be all of your mouse RNA genes that we map to human, and then human, obviously the human. You're going to get outputs that we will put through some machine learning and post-processing algorithms, but it's getting exciting. So everybody is on the BioInnovation platform. We want to go to that, and I'll go along with you, or attempt to, as we're going through this.
This is a demonstration of Wirecast.
Okay. So if you're not yet signed in, we have a sheet that has to cross that table, as well as in email. So the login is test@biotech for the username, and then the password is a bunch of letters and characters. So you can either find it on the sheet or via the email. I don't know that memory. And just let me know when everybody's logged in. And for those of you that are already logged in,
This is a demonstration of Wirecast.
unless I change it.
We'll probably change it after this.
Yeah. But if you're wanting to, look through the platform, or you want to make it more.
Yeah, the only problem with this account is it's one account for every different. So there's no data gets confused. So that's what it is.
This is a demonstration of Wirecast. This is a demonstration of Wirecast. This is a demonstration of Wirecast.
False.
This is a demonstration of Wirecast.
All right, so I'm going to go ahead and, for those that are a little bit behind, just grab any of the people that are associated with me and they can help you. Okay, so for those of you that are there, you should see this page. Everybody sees it? Yeah? Okay. So we're going to go to RNA-seq underneath transcriptomics here. A single select should do it.
This is a demonstration of Wirecast.
Great. Okay. So I talked a bit about formats already. Essentially, before you actually build your pipeline, you need to give some inputs to the system so that it knows the type of format and what it's actually going to be analyzing. So the first, for NGS, we have FASTQ files, not FASTA, so we'll leave that the same. For organism, if you go to the drop-down, we are mapping to a human and a mouse, and that's at the very bottom here. So we'll select that.
This is a demonstration of Webcast.
It's a brand new species. We just created it. And then our reads, Claudia talked a little bit about the difference between single end and paired end. The samples we have are paired end, so we want to do this drop down and select PE. What does this mean here? Essentially, we have the reference genomes already inputted into the system, so that's just all that's saying. Once you have this set, now we're ready to select upload files and add groups.
This is a demonstration
and if you
of
look up at me,
Webcast.
that's kind of how I know you guys are good for you to keep moving. Okay. So we could upload the FASTQ files, but those are large, and it would take more time than this module just to upload them. So we're going to not do that. We're going to go to this URL, bulk upload here, and we're going to upload that SVL file. So let me just show you the inside of one of these files. Yep, that is it.
This is a demonstration.
So when
of
you select that
Webcast.
in the URL upload, essentially what this is all of those FASTQ files are loaded or saved in our server, and these are links to those individual data sets. Yeah, great. So I will hit bulk upload, select the SVL, everybody's good, and choose. This will load to 100%, and now we need to define groups. So what you'll see is boxes representing each individual sample. In that sample are 2 reads since it's paired in, so that's why you see both lines. RNA-seq has 2 main options. You can either build gene expression tables, which is what we're going to do today, or you could do differential expression. If you already know some information about your groups and you want to try comparing those. Since we're doing just gene expression, we're actually going to drag all of these boxes just into group A. But if we were doing differential expression, you have the option to add more boxes and make more groups. No, it will not. It will not matter. You can pick the very top, go to the very bottom. Yeah. No, it won't matter. This is only important when you're doing some type of differential expression and you want to check it.
This is a demonstration of Webcast. This is a demonstration of Webcast.
Now drag all of these into the box at the very bottom. Yeah. I got a lot of
This is a demonstration of Webcast.
Everybody's looking at me a little bit. So, okay, once we have those groups loaded, we will select continue. So now your groups have been added. We can actually start building a pipeline. How do we know we can start building a pipeline? You can see that the start has darkened and has yellow haze around it. When we start building the pipeline, you'll notice that as well. So if I select start, a pop-up will come. And this is for all of the pipelines that you build in the
This
pipeline.
is a
The idea
demonstration
is that it's
of
supposed to be
Webcast.
interactive, helping the user figure out why they might select an individual algorithm. For the start, this is just a logical flow for doing an RNA-seq experiment. That would include steps like error correction, mapping our genome, mapping our exon junctions, forming your isoforms, and then either building expression tables like we'll do today, or differential expression. Okay. So,
This is a demonstration
So,
of Wirecast.
if I scroll down, right, you'll see, oh, I cannot scroll down, but that's fine. Oh, the window is just low. Sorry, guys. Okay. My bad. Yeah, so the Tau of Science means that it was developed at the Tower Institute in Israel, which is where this entire platform was originally created. And all of the ones that do not have that are open source algorithms. So the Top Hat, Bowtie, these are very common algorithms that you can use on other platforms like Galaxy.
This is a demonstration of Wirecast.
You don't have to run Top Hat and then get an output that's a format that can't go into the next algorithm. This will do all of that for you as you select it. Everyone see this page? Great. So now we want to build the pipeline. You've seen a few images of it already, but we're going to select Bowtie 2 for our mapping. You'll select once. You'll see a drop down or pop up. That pop up is going to have a little bit of information about how that particular algorithm works and a link to the original publication.
This is a demonstration of
Once
Wirecast.
you have selected, you'll just select Save. And as you can see, we now have a line going from story to Bowtie. What you might notice is that once I actually selected Bowtie, a lot of those algorithms or the buttons that were lit up are no longer. And that's because we cannot choose those anymore. So this helps the user not build a pipeline that wouldn't work. Okay. Next, we will select RSAM. Similar concept, a little bit of information about the algorithm.
Of Wirecast.
Once those are selected, this is our entire pipeline that we are going to be building, so we will hit End. You'll hit Save on the parameters. It will load, and now we want to name it. Elia mentioned already, you're all on the same account. So if you want to come back to see your pipeline, I'd recommend including your name in the name of your pipeline. And I'll do the same.
This is a demonstration of Wirecast.
Once you've named it, select Save. And then we want to select Run Pipeline at the top. Now, unfortunately for you guys, this won't finish in the time that we have. So I've already given you the TXT file.
This is a demonstration of Wirecast.
We have to recognize that a human genome and a mouse genome are large, as well as we have tons of RNA fragments that all need to get mapped. So this will finish quickly. You could probably check it by the end of the day today, but it will not finish within this quick module. So I'm going to go back to the PowerPoint for just a bit. Try to go back to the PowerPoint. And I'm going to show you what an output would look like. So if you say this afternoon, have some free time, and you want to look at what the pipeline has done, it's going to look like this. So you're going to get the job outputs and all output files. You would select all output files and then select download. And then that's going to give you those tables that we already talked about, your expression gene tables and your expression isoforms tables. And what you have
is a demonstration of Wirecast.
If I open that table, what would it look like? So the first column are going to be gene names. And this is a little bit different than what you might be familiar with. Often when someone says a gene name, if I was talking about estrogen receptor, you might just see ESR1, right? This is the NCBI or ensemble number that corresponds to that individual gene. Across, you'll see your individual sample names. We saw those earlier.
This is a demonstration of Wirecast.
Here is the actual expression, so the number of RNA fragments that match those individual genes on the genome. And have it's before normalization. This is important. We recommend that you go to our machine learning section, which I have here, and actually run content normalization. Why? The raw data sets often have some type of unwanted technical variation. if you did not normalize your data,
This
then
is
you
a
need to take this
demonstration
table
of
and
Webcast.
move all the way on to gene ontology and make some decisions about biological decisions that would be wrong because of characterization. So it only takes a minute or two to run. You would take your table from that to awesome, find the content normalization, and then your table would be normalized. Now it's going to be actually right, because the RNA gets fragmented.
This is a demonstration of Webcast.
So this is the table that you do have. And something I'm going to just show you that we're going to do today, if I took this ENSG gene number, I want to know what it is, because those numbers don't really mean anything to me. Then I can put it in different biological types. This is an example of an ensemble. So MMP14, I now know it's a matrix metalloprotease. What would be
This is a
important
demonstration
to a
of
biologist
Webcast.
might be something like the location of it on the chromosome, or what transcripts are related to that gene. I could then take those transcript numbers and look at it in the isoform. And then have these two here. And we did give you these links, so we're going to give you these. So here you have ensemble and David. Has anybody used these before? One got okay, good. So ensemble is great for many things as far as trying to understand the biological.
This is a demonstration of Webcast.
So it can annotate genes, it can do multiple sequence alignments. David can do a lot of this in the ensemble, but it also can give you biological pathways. So if you take in your table, which we have now, the ones that we work with often have 60,000, 100,000 genes. Once you're able to sort through that and identify those that are important, you can put them in David and get some kind of biological pathway. Alright, so we're going to go back to work now.
This is a demonstration of Webcast.
We're going to go ahead and actually take that pipeline and look and make a scatter plot to see the results. Then use David and ensemble to have a biological meaning. I'm going to talk to you about some other machine learning techniques and then give you an idea of what an actual RNA project would look like. So let's go back to the T-Bio platform.
This is a demonstration of Webcast.
Yeah, you don't have to be connected to Wi-Fi for it to be running, but if you wanted to get the data, you obviously site for that.
So it's your servers that are running.
An option for that?
Yeah.
This is a demonstration of Webcast.
So we've had researchers that have their own algorithms or their own ways of doing things. We can integrate that in. That's possible, but it's so complicated somehow. So everybody's here? Yes? Okay, so we want to build a PCA pipeline. The way we will do that is going to areas of analysis, then drop down to data integration and modeling, and then we're going to choose machine learning.
This is a demonstration of Webcast.
Sorry. And now is when you want to make sure you have that .csv file. So that we understand the connections, that .csv file is
This is a demonstration of Webcast.
And what we want to do now is run principal component analysis and see if we can identify any separation of groups between these samples. So everybody's here? Okay, great. So we're going to go to upload files. And instead of doing a .svl, we're doing a .txt, so we will go to upload files. The name of it should be breast human RNA-seq expression table. Table? Yeah. Okay. So we will choose. Once that uploads, we'll select continue. Okay. So now that we've uploaded our file, start is an option that we are able to choose. So we'll select start. You'll get a pop-up just talking a bit about the machine learning pipelines that are an option. And we want to choose PCA. And it's the first algorithm
demonstration of Webcast.
Okay. This will tell you a bit about PCA. And there's a link here that's kind of a dumbed-down version, the only one I understand, that explains what PCA is doing and how that works. And Claudia's talked about it a bit already. So for parameters, it is already logged. We would select yes. Before we did do normalization, but that is normalized, so it is logged values. And then column start and end from. So column data start, it says 2.
This is a demonstration
right?
of Webcast.
Column is the list of genes. So we are including that because that's not actually what our name is. So we start from 2. We have 21 samples plus that column with genes, so then the column in data on would be 22. Once you've hit save, select end, select save again. And I would say just kind of go with the, yes? Okay, go with the same idea, put your name there, add something like PCA and whatever your name is. And then select save and run Python. So principal component analysis can run in a minute or two. So what we're going to do while we're finished is look at the visualization links. We have a visualization link for genes PCA as well as a link for PCA samples. So you may have to reload them when you go to it. I will have to as well. And for any of you that don't have the links already open, just kind of look at us and
This is a demonstration of Webcast. This is a demonstration of Webcast.
This is the first one. That's it. You have about half the room there. Yeah. Okay. So I'm just going to talk about the results from this. So what you have demonstrated here is the table from the PCA result. What you get that we've inputted is just the first 2 components, so PC component 1 and
this is a demonstration of Webcast.
The reason is that those explain the most amount of variance across the samples. So what you're looking at here, each individual point represents a sample. And the principal component analysis has reduced all of those genes that are represented in each sample to a simple component, PC1 and PC2. And if we look at it, we see separation. It's a little hard from the back to understand, but you can see blue samples for the triple negative,
of Webcast.
green for the ER, and they're grouped together separately. And then there's one middle sample, which is HERT2, that's purple right there. And it doesn't really separate between those 2 groups, and that's expected. If I take this PCA of samples, and now I want to look at the individual genes that are represented by each sample, we'll look at the next visualization, which is the other scatter plot here.
This is a demonstration of Wirecast.
Yeah, perfect. Cool. This is interactive. So each, as I said, each individual point represents a gene. And so if I select that point, I can now see the expression profile. And what I mean by that is I took the table and looked at the amount of RNA that mapped through each of the samples.
This is a demonstration of Wirecast.
ENSG 09131. And you can see that it's upregulated in estrogen receptor cancer, and then downregulated the amount of RNA that's being produced in triple negative and in that HER2 sample. And that's interesting. That is biologically interesting because there has to be a reason as to why we have that difference in expression. If we go back to just this PCA table, and this isn't the same for all of them, but the reason you're getting this spread of dots is that those that are closest in the middle, for the most part, have either low expression or not many of the samples had RNA for that gene. As you span out, we have expression across the entire set of samples. Now, I'm going to test this. I do not know if everyone's PCA will be finished, but we can go ahead and look. But you can play around with that visualization for a minute, and I'll just see. Yeah, for the most part, they do.
This is
cool.
a demonstration of Wirecast.
So, somebody had a little trouble with making it, and that's okay. We can just share. If you go back to the BioInnovation platform, you're going to want to select your name, and how I got there, is selecting my pipelines here. And you can then find your individual run. And I might pick someone else's. Let's just see. Let's see if Ben's is any good.
Demonstration of Wirecast.
Okay. Everyone's following? We're good? Okay. So you'll select that pipeline. And then now we have job outputs and all outputs, as I already demoed before. We'll select the all outputs section. And then select download. Once you're there, you'll see a number of files. The one that we're interested in is the actual PCA result. A number of these files are either what you've inputted or like a log, just talking about how that algorithm ran. So we are selecting this one here, the second on the left. And you want to download it, and ideally, if you can, open it in Excel. That will be your next step. Already.
This is a demonstration of Wirecast.
So the percentages go across for each of the individual samples is the number of columns in here, and the percentage is the amount.
This is a demonstration of Wirecast.
Across all of the samples. So it's reducing each of those to one dot, and so there's amounts of variance, a difference between each of those individual samples, and so it goes in order. PC1 and PC2, and then it's one number. So the difference is just say how much is actually exploded by that PC. Anybody have a view of that?
This is a demonstration of Wirecast.
It's the amount of variance.
Yeah. Okay. So higher.
So, the higher the better.
That's why you were saying that you made those plots with those two, because those have the high variance.
Right. Ideally, we want an even higher variance number than what we're getting here. So if you do PCH later, and you see 16%, you probably don't have the best, you might want to go back. but we made an educational data set.
This is a demonstration of Wirecast.
So it's just a workshop project. So once you have the table open, we're, as we said, focusing on PC1 and PC2. You'll just highlight over them, not including the percentage numbers, so just the coordinates for each of the individual genes. Then go to, no, ignore me. Then go to Insert, and we want to build a scatter plot.
This is a demonstration of Wirecast.
So as you can see on mine, I've only highlighted from 2 to 22.
This is a demonstration
So once you have that,
of Wirecast.
It should look like this. Does it for everyone? Similar at least? I hope, yeah? And if we go back and we take a look at this image, and then we look at what we have for the visualization of, well, we'll reload it. It is the same. The difference is just yours hasn't been individually labeled for the samples, but you can see a separation between us. Something that's interesting that you can do, we won't today, but you can go back to this table. You don't have to select PC1 and PC2. You could select others to see if there's a different separation by something else. What you get from the PCA is an explanation of some type of biological meaning. So with, I'll go back to this, that's actually labeled. So with this, I was able to see as I'm going through the project that there's separation between one breast cancer subtype and another breast cancer subtype. And since PCA is looking at the amount of variance,
this is a demonstration of Wirecast.
There's a number of genes in that expression table that are different between these subtypes. And I want to identify what those are and try and understand the mechanism behind it. So we're going to do that just a little bit. We want to go back to our Excel and open, sorry guys, the file that we uploaded originally. So we're going to open that .txt file, and I'll go back over what the name and stuff is as we do that. So file,
this is
open.
A demonstration of Wirecast.
That's, nope. Yes. So yeah, breast cancer RNA expression. Nope, no, still for me. Table.txt. So this is what you're going to open in Excel.
This is a demonstration of Wirecast.
You have an ensemble open? So you saw this from the PowerPoint, and you're welcome to take this and start interacting with it if you want. But what we're going to do is select that first gene, and then we're going to take it and copy and paste that, and we're going to look at it in ensemble. Let's see if we can figure out what this gene is. Now I'm going to copy. Oh, I may have already looked one up, so we're going to... here. Okay, there we go. So, everyone's on ensemble? Yeah? Alright, so you're going to paste that gene into the search engine, and you can go ahead and
this is a demonstration of Wirecast.
Okay. So we get a lot of search results back, but because Ensemble is quite competent, the first one is what we actually want to select. You can see that the gene matches, so the gene number matches what our search query was, and it says human gene. And we know it's a human gene, this is something I didn't have to explain, by the first 4 letters. So in the large table, we know that it's a human gene by
This
EMS
is a demonstration
but
of
if I was
Wirecast.
looking at mouse, it would actually be EMS gene, MU, for the mouse genome. So let's select that. Okay, so some volunteers can start telling me what is the descriptor for this. I will call.
A different page up on the
Okay.
Like says transcript. You want to know the description?
Yeah, let's see.
This is a demonstration of Wirecast.
Right, we wouldn't have known that if we hadn't searched it, right? We wouldn't know that by the number. And as I said before, you get a lot of interesting results from this. You can find the corresponding transcripts, or isoforms is probably another common name for that. You can see that there's 6 of them associated with it, which is definitely interesting, and that some of them are not actually protein coding, which gives you a little bit of an idea for biology what isoforms would be important to look forward at. The chromosome location,
This is a
you can scroll
demonstration
down
of
and see like, the location
Wirecast.
of it. I'll scroll down for you. Location of it actually on the chromosome, what's protein coding. Something else you can do, let's see, how much time do we have? Yeah, we have a little time. Something else you can do is also look at this in David, but it's better to do with a group of genes than it is just with a single. Great. Okay. So I'm going to talk about a few of those genes that you're seeing in that table. And then you yourself could go back and actually do the same thing that I've done with the table that you have. So we want to define those breast cancer receptors. And there's a ton of genes when we do other machine learning techniques like BioInnovation that differ really significantly in their expression profile between the estrogen receptor and the triple negative. But you can't only look at the expression profile. You now have to understand the biological reasoning and then pick those genes that actually
Wirecast.
So I'm going to go over a few, and you want to pay attention because there's a little bit of a quiz at the end. Okay. So why would matrix metalloprotease be upregulated in triple negative cancer and not ER? There's a few answers to that, but why is NMP14 even interesting? So it's expressed outside of the cell, and it's essentially attached to a little receptor, and everything that interacts with it starts getting cleaved or activated. So whenever a cell expresses NMP, it's able to degrade an extracellular matrix. What does that allow a cell to do? It allows it to move, it allows it to become metastatic, and that would be very important if you have a type of breast cancer, that your breast cancer is overexpressing this gene, which means it's overexpressing that protein, and that it has a high likelihood to become metastatic and move from one part of the body to another. I have here too a welcome to gene resistance.
This is a demonstration of
Wirecast. If you wanted to
Wirecast.
take the table later, you could go to this database and actually input every single gene. And you can see if anybody has ever published anything on breast cancer related to those genes. And here I have 3 of the clinical data sets that have been posted showing that this cancer type is differentially regulated in future breast cancer subtypes. So what do we learn from matrix metalloprotease? It is upregulated in triple negative cancer and downregulated in estrogen.
This is a demonstration of Wirecast.
I'm going to do 4 of these genes, and then we're going to so TFF3, or tree pull factor, is extremely downregulated in triple negative and upregulated in estrogen receptor. I put here that it's an estrogen-regulated protein, and I keep talking about these 2 hormones, but they're really important for breast cancer. Triple negative doesn't have that receptor, so all of its associated genes don't get upregulated. So its role in estrogen positive cancer is actually to upregulate other pathways such as STAT3 and induce angiogenesis, which is when blood vessels are brought to a tumor. It allows it to grow faster with more access to blood. And this is important because it's also a growth promoting factor. Okay. We talked about estrogen, so we can't skip it. You can see that estrogen receptor positive cancer is upregulated.
This is a demonstration of Webcast.
But there are still some expression, and I think that's an important point to make. We're looking at RNA. When the doctor looks at it, he's normally looking at the protein. And normally, what's happening here is that there's mutations in this gene or in this RNA that just make it where it does not result in a protein. ER-positive cancer has tons of estrogen receptors on the outside of it, and so it takes in this estrogen, and that estrogen binds to certain genes.
This is a demonstration of Webcast.
Last one. Progesterone is incredibly interesting to me because typically in estrogen receptor positive cancer, the progesterone receptors are also present. But if you take in, if that cell takes in a lot of progesterone, it's going to slow down the growth rather than quicken. So progesterone can put a brake on the division of breast cancer cells, but we often see other subtypes, such as breast cancer that does not have progesterone expressed, and other variations of that sort. And you'll notice that HER2 has absolutely no progesterone. But let's see. All right. If I am a researcher and I somehow lost all of the information from my data sets except for the RNA, I know that part. I don't know anything about the names. I don't have anything.
This is a demonstration of Webcast.
Can I figure out the subtype? The answer is yes. So let's see if we can. So your first question is you have an expression profile with a high estrogen receptor, estrogen. We have a high progesterone and a low matrix metabolic release 14. We have three options. So the triple negative, the estrogen positive, or is it HER2? And anybody can talk, except for those that are looking.
This is a demonstration of Webcast.
Yes. Look at this. Doing better than they might. All right. Second profile. We have low estrogen, no progesterone receptor, and high matrix metabolic release 14. This is where it gets a little hard. It's not really. All right, guys. And then obviously with last one, low estrogen, no progesterone.
This is a demonstration of Webcast.
Okay. So I don't want to overwhelm you with machine learning stuff, but I do want to just go over one other that's possible. And you have a link for this. It's your last visualization link, so you're going to want to go to that. Factor regression analysis is similar to PCA. It allows us to define groups, but in factor analysis, you're giving it input. With PCA, we didn't tell it anything about the specific sample or any of the genes. It just separated by variance.
This is a demonstration of
With factor regression
Webcast.
analysis, we are giving it inputs. Yeah. With factor regression analysis, we are giving it inputs. The inputs that we gave were to compare the triple negative and the estrogen receptor cancers. And then the second input that we gave, which we haven't talked much about, and that's okay, was to compare the different types of mice. Our results did not give us very many genes that differed between mice, so I went ahead and took that out for this. And here we can actually see a list of genes that we know have an influence in either one or the other groups. And I'll go to the visualization itself. Yes. Should. Is everybody on it? I might not be.
This
on that page.
Demonstration of Webcast.
Yeah. So the same page we put those. Yeah, she's got it.
Do you have colorblind ready plotting options?
We do not. That's something we should keep.
No, I'm not, but some publications require it.
Yeah. So this has the same gene expression table that you've seen or already opened in Excel. The difference with factor analysis is that we get a lot of additional new columns, such as t-test and f-test. If you know what that is, great. If you don't, look it up later. But we sort through these, picking statistically significant numbers, and we're able to identify a group of genes that are really playing a role between these two factors. And you can go through this and see the results, because this actually is our factor analysis results. Okay. Let me put your gene.
This is a demonstration of Webcast.
And so you can always go back to that if you want to look at, you're entering a particular gene, see if it's in the factor analysis video. What we did today is a really small example of what a researcher would actually do when they're getting their data sets. And I've outlined it a little bit here. So if I was, which I am, if I was a biologist and designing an experiment, the first thing I'd want to do is actually figure out what my question is. What is my hypothesis? How do I design it so that it's best for Illumina sequencing, perhaps, or just for a sequencer itself? So you would take those samples that you produce over weeks or months and extract the RNA. The RNA then gets fragmented. You reduce the amount of ribosomal RNA, and then once you've done that, you're going to run that in a sequencer of any sort. And then once you've done that, you now have your samples. And you do a little bit about what we talked today. So you would do a secondary analysis that includes mapping. A lot of times researchers are going to pick multiple algorithms. You saw a number of mapping algorithms on our platform, and they would compare those results. They may do a number of error correction or post-processing or pre-processing before they're actually getting started with that mapping. So there's a lot of steps that we skipped over today that they would normally take. Finding isoforms, building the transcriptome, and then just cleaning up the data. Another thing that we already did for you is when you get these tables, there's actually a lot of genes that don't have any expression.
Of Webcast.
So we clean those out before we start normalizing, otherwise it would affect the results. Then the third part, the fun part, is the tertiary analysis. You would do a number of machine learning algorithms. I showed you an image of all the ones that we have underneath PCA. For example, you have by association results here, different clustering results. Using those different machine learning techniques, you're able to get down to an even smaller number of
This is a
genes
demonstration
that
of
are affecting
Webcast.
some type of biological role. And then my favorite, the gene summary and ontology report. Once you've done tons of work, and you've got those lists of genes that you know are playing a role, this is when you would start figuring out what they actually are and what they do. And we play a role in all of the secondary analysis and a lot of the tertiary analysis as a company. So thank you for listening. If you have questions, you can ask me now or you can ask me later. And then I listed a bunch of links, most of which you already have, in our contact info. Thank you for coming with us.
Yes. Yeah. The evaluation.
We have.
It'll be right now.
Okay.
Please.
If you haven't, yeah. So if you haven't taken the evaluation, I know 5 of you did. Thank you so much. If you haven't taken it, it would be great to take that just so we have it, and then we'll send you a post-evaluation as well. Just input and please be honest. Not nice.
This is a demonstration of Webcast.
Okay, great. Thank you so much. Yeah. Any questions or? Yeah.
So what other types of analysis do you do besides RNA? So
we showed that in the very beginning, but I'll go back through it. No, you're good. You've been in and out. Yeah. I won't tell you.
No, I already know.
Yeah, we see. So NGS data sets such as transcriptomics, genomics, de novo assembly. We have a proteomics pipeline as well, libraries of small molecules, and then some data integration and modeling, such as virology and machine learning, which you saw already.
What do you have for biopolymer structure?
So library of small molecules, do you want to show them there? Because
Okay, so the platform itself covers many different types of data, and the main differences are by the input data. So next generation sequencing, mass spec, structural biology. In structural biology, what we have is a number of sections that we're working on.
Yeah.
Essentially, if you go there,
This one.
yeah, this is the section that is already developed. Others we're still working on, but this one takes the structural data, transforms it into linear data, and then you can either query a database and find similar ones or opposite symmetries, so you find possibly they match in a different way.
This is a demonstration of Webcast.
By some type of group. So essentially, you're looking at, if you're talking about toxicity, for example, you might identify those that are more toxic, less toxic, or have other physical chemical properties. So that's the basic idea here. What we actually a lot of times work on is integration and custom workflows. So a good example is a virology pipeline because it actually takes many different types of data and integrates them. So you go through SERSeq data. SERSeq data is a specialized RNA preparation process for viral RNA. That viral RNA has low frequency mutations, and that's what's important here. So to detect those, the SERSeq method was developed. Then you want to correlate that with the host response. So you want to see which mutations, for example, correspond with a specific biological function of the host, like immune response or other things. And you might then want to identify specific areas on the viral proteins that could be potentially therapeutic targets, for example. So just to give you an idea of what that would look like as an output, this node. So just as an example, you have a comparison of dengue virus that we're developing in human and mosquito. And what we wanted to compare here is mutations that are opposite.
This is a demonstration
of
Webcast
with a specific fitness. A fitness corresponds to how much that that specific mutation goes up or down. If it's bad fitness goes down, good fitness goes up. And so the good are the beneficial, detrimental are the bad ones, and we're interested in the opposites between the two. So if you select here, for example, the opposites, what you would see is an example like this, right? It's detrimental in human and beneficial in mosquito. So that's interesting to us, and we actually are, might be interested in a position of where that's located. So what I can do is I can go through a few of these, select them, and then I can have an output of individual point mutations, their fitness properties, then I might want to look at molecules that might fit into that pocket, into that protein. So that's the type of workflow that the system was designed to tackle. And so as you can see here, kind of all of the stages that you saw in the visual format, right? This is how you get the different data sets. That small molecule section is also integrated. Any other questions? So if you have any other questions, if you've done this before and this was kind of boring for you, you can contact us and we can talk about other things. There's a lot of open source stuff about this. You can definitely learn. Our goal is not to compete with what's open source. Our goal is to, first of all, make it accessible to a wider group of people. Make sure that it's simple enough that anybody can get biological meaning out of it, not just the table. And also work on these custom challenges that I think require a collaboration between the biologist that designs the experiment, that has innovative methods, people with data, other types of data. For example, in the clinical space, you might want to have clinical records. So you don't just work on phenotypes, you also correlate it to some kind of clinical history, for example. And then you want to work with interpretation, right? So what is the biological function that might be the underlying reason for that correlation?
Do you have an exclusive license for the Tulane C3 operation?
Yeah. Yeah, so the platform Like we mentioned, the tau are all the algorithms that were developed at Tau Bioinformatics Research Center.
Of Webcast.
These are just, if you go through all of the different areas of analysis, some things we're actively working on, some things are more ready. So transcriptomics is a more assembled and tested kind of pipeline. Other things are still in development. So what you will see is, in many different places, you will see something that actually is just starting to, take shape, and some of the buttons are empty, and that means we're planning something. We're still building.
Demonstration of
Webcast. So, just to kind of give you, I know that some of you actually do your own algorithm development, and so let me just log out of this test account. So yeah, so this is essentially the back end of the system. And the back end of the system allows the developer to add on a module into one of those pipelines to define the inputs and the outputs, connect it into a job pattern.
This is a demonstration of Webcast.
You were referring to, right? So you could add your own script that, for example, transforms a table in a specific way, prepares it for a different algorithm. And then you can do all kinds of things. Known methods, visualization that we showed you, that can become a button or other things. Yeah, so right now we're also actually discussing what's the best way forward. This is a working prototype.
This is a
We
demonstration
use
of
it
Webcast.
On small-scale projects or for our own research. So the end goal is to do cloud, hybrid cloud, and HPC. So essentially, some algorithms are very RAM intensive. Some of them are not. Some of them work with proprietary data sets. Some of them work only with de-identified data sets. So that's a way that we're thinking to distribute the different algorithms and the resource allocation. So, but that's just a concept right now. We've tried, Azure and Amazon and things like that, and there's limitations and benefits, and so we think that hybrid cloud should be the ideal thing going forward, but we haven't figured it out completely. Yep, thank you.
This is a demonstration of Webcast.
I'm not a Reds fan, but
This